Hi everyone. Welcome to this week's Single Cell Exploration Club. My name is Alexis Garduno. I'm one of biotrained scientists. Today's sort of weekly session will focus on tissue residency versus cytotoxicity, and really being able to explore these different CD4 positive T cell states in atopic dermatitis skin. So this will be explored through the new Talk2Data, which will also incorporate BBrowserX Pro for the more advanced analytical analysis, and really how you can harness the public database to really answer these specific questions within this specific disease case study. So as I mentioned, most of these questions will really relate to how Talk2Data can turn a specific biological question into reproducible cohorts, whether that's through selective disease filtering, tissue, sampling site, and cell types. From there, we'll go through our new AI Sonya agentic model, and really how we can use this model to look at other specific questions relating to CD4 sort of T cell states in this disease. And if we're able to really look at and connect different cell states and really how these cells have local signaling as well as therapy context. So this will be sort of the main focus. The agenda that we'll sort of go through will be sort of an intro first on really inside sort of what we have in atopic dermatitis lesions, what is the pathobiology and also therapeutic gaps. From there, we'll go to sort of an intro of what Talk2Data is for anyone who hasn't really used that module in the system and wants to get a bit more information on that and how it harnesses cross-study analysis. We'll go through some specific questions as well. For example, how do these states shift depending on whether your gene is located within lesional versus adjacent normal tissue, and does that sort of signaling shape sort of local signaling? So we'll look particularly on samples relating to atopic dermatitis across multiple public studies. We'll try to sort of show you how you can look at very specific cell populations and also specific sampling site tissue structures. So that will be sort of the first part. Within the AI Sonya, we'll try to really look at how we can prompt the system with very specific questions relating to lesion versus adjacent expression. Can we quantify these different subset proportions within these different sort of cell types? And can we look at Pearson correlation scores as well? So that will be sort of the second part. And then we can then direct that filtering and those analysis outputs and really look at one particular individual study within BBrowserX Pro to take sort of this question even further. How can we look at specific cytotoxic or resident memory T cells and how they actually communicate, and do we identify any ligand receptor shifts? So this will be something that we'll map within BBrowserX Pro. Okay. So a bit of an introduction or sort of really how we understand what's sort of happening inside the atopic dermatitis lesion. So these lesions are not usually uniform. You don't really see a uniform inflammation. They are very site-specific ecosystems that are usually dominated by these sort of two different CD4 states, which are these tissue resident memory T cells, and really how these sort of cells are anchored by specific genes, which we'll sort of cover today, which are CD69. You also see some expression of ITGAE. You see some suppression markers, and we see other sort of cell state CD4 T cells, which are the cytotoxic T cells. And these cells, really what we find is that they express very particular markers like PRF1, GZMB, and other sort of cytotoxic or effector markers. So we'll try to sort of uncover if that's something that we can really see across multiple data sets. Can we look at not only sort of the lesion sites or sites that are sort of diseased tissue, but can we compare that to adjacent normal tissue within the same patients? So that's questions that we can answer through the new Talk2Data. and really try to understand this complex pathogenesis within these different autoreactive T cells, and really mechanisms that are still unknown to this day. So that's something that we'll try to really uncover. Before I go into sort of the example or the actual live demo on how you can perform these steps in the system, there are certain things that you'll need to really understand. So within the BioTrain system, when you go and access, we do have the Talk2Data sort of module, which is sort of our main repository where we curate all of the public data from the system. And the way that we do this is that in order for you to be able to run cross-study analysis, you need to be able to understand how our team actually annotates these cell populations, how do they standardize, and how we manually curate not only the sort of expression matrix files or the studies, but we go through each individual data set. We look at these supplementary files, and we specifically try to replicate what sort of specific markers are sort of used to label specific T cell subtypes, and can we actually sort of replicate and validate these results in order to create these metadata terms within the system. So these are things that you'll sort of notice when we're exploring the data, how we actually standardize the ontology and how we're still able to keep the original sort of cell type granularity as well. Okay. We do standardize across different sort of conditions or different labels. So not only the sort of cell ontology, but also disease ontology. If we're looking at different anatomical structures, if we want to compare this across different sampling sites or different treatments, we need to make sure that these are ontologies that have been used or are specific databases that we can sort of utilize to be able to map across every public data set. Okay, so something to keep in mind as well is that the new Talk2Data system has been sort of expanding throughout the years. Currently, when we're only focusing on human data, we have close to 3,500 studies or data sets and over sort of 200 millions of cells. That's only encompassing the human sort of species, but we do have multiple layers of the data, including different databases. So not only single cell RNA-seq, but also spatial imaging data, bulk RNA-seq data, and really looking at different versions of the data. So not only studies that we particularly curated in the database, but also other publicly sourced atlases that you can access as well from the Talk2Data system. Okay, so this is just an example on how you can filter and really look at different species as well. You can get an idea how our sort of agentic model is sort of processing the data. So where is that data coming from? How many cells or how many number of data sets are being added to that input, or different inputs that are allowing you to sort of run specific sort of queries, and the system would be able to analyze those biological questions through natural language. So these are sort of databases that the system is really pulling to be able to integrate within our AI agent. Other things to keep in mind is that when we are sort of discussing spatial imaging data that we've curated from the publicly available studies, we not only concentrate on one specific technology, but we do integrate many different technologies across different types of either sequencing-based, proteomic analysis, or other imaging-based technologies. So these are studies that really you can explore at a deeper scale, and you can get these different layers of the data to really sort of answer your specific biological question. Okay, so this is something that we'll cover really. How can we utilize Talk2Data to add a search? How can we filter studies? And how can we base our filter on very specific multiple research criteria? And in this specific use case, what we really want to try to understand is sort of the lesion biology within atopic dermatitis. Can we really distinguish different CD4 positive T cells, and particularly markers that are seen in tissue resident memory T cells, as opposed to what you would see in cytotoxic T cells within these lesions. So in order to do this, we'll go into the Talk2Data system. You can access the repository by going directly into the Talk2Data module. From there, we'll keep the single cell database. So we're particularly focusing on single cell for now. We'll keep the human database, and we'll also keep the current AI Sonya, which is all the studies that our team have imported into this current version of the database. Any other sort of publicly available sourced atlases can also be accessed in this case. So from here, our first really question that we really are trying to understand, and if we go back to what that is from the agenda, is are we able to really look at these different shifts within lesional and adjacent normal tissue? And can we particularly look at genes that shape local signaling in atopic dermatitis? So if we want to really understand that question, we'll go into the system and we'll try to sort of add these specific filters to address that criteria. So the first thing is that you want to have a anatomical structure filter in order to make sure that the samples are coming from specific areas that you're trying to focus on. So in this case, skin of the body is the standardized ontology where it will include specific sample or lesion sites within the skin tissue. So I'm going to apply that first search, and you'll see that it automatically adds that first sort of criteria. The second thing that we want to add is sort of the cell types that we're trying to really target within the database. And in this case, we're really looking at the cytotoxic T cell populations as well as these regulatory memory T cells. So in order to do that, we'll add that in the system. You'll be able to see that there are different options when it comes to cell ontology. So either the major or the hierarchical sort of labeling, but then you'll get to other labels that are more granular, like cell subtype populations within sort of a higher level. Sort of ontology. So that's something that you can add. For example, if we want to really look at cytotoxic T cells as a whole, or we want to look at CD4 positive alpha beta memory T cells, we can add those as well. So here in CD4 positive, we want to make sure that we add the memory T cells. We can also add other granular memory T cell groups, for example, regulatory memory T cells that might have different cell state shifts and might be expressing these specific targets at a different sort of level based on whether this is lesional sites or the adjacent normal tissue. So that's something that we can add as well to really look at that shift. And then other sort of populations that are cytotoxic as well, which are the CD8 alpha beta cytotoxic T cells. So these are sort of our cell types of filters that will give us some indication as to how our targets are differentially expressed within what context or what type of sort of local expression. Now, in order to be able to filter within the database and particularly only look at specific disease models, this is where we'll add the atopic dermatitis, which is our main sort of use case for this session. And we want to make sure that when we're running this analysis, we're including both disease tissue and adjacent normal tissue for comparison. So since we want to really look at local expression within the lesional sites, we can start by adding in the sampling site disease tissue. And then we can sort of progress into adding the adjacent normal tissue after. So at the moment, what I really want to look at is very specific markers that I can really determine that these are specific for cytotoxic T cells. Some markers might be more specific to these sort of tissue resident memory T cells, and I want to be able to run this across the database. So you can see here that we have currently about 7 studies that particularly fit our criteria, both at the tissue structure level, at the cell type specific level, and also studies that would have information relating to the sampling site. So studies that we can compare both disease tissue and adjacent normal tissue, and also other information that might be important, like differences in treatment response or specific agents that are used as well. So these are things that really harness and provide this information for you to answer these biological questions. Now that we have our specific sort of search criteria, this is where we can proceed to the next step. So how can we go into the actual analysis tab? And this is where I can go here into analysis. Okay. So if I go into the analysis tab, you'll see that there are specific targets of interest that I want to really sort of look at. You can add other genes as well within the system, especially if you're focusing on multiple targets or like the top 50 targets, and you want to sort of identify any off-target effects across other immune cells. You can add these sort of genes within the search as well. So now that we're within the system, we can actually look at a sort of bubble heat map of this sort of expression. And we can look at this within sort of our, I guess, our criteria or our filter criteria. So here within the skin body structure, we have these different cell populations of interest, the cytotoxic populations and the memory regulatory T cells or other sort of memory, sort of resident sort of T cell groups. So what we can see initially from the expression is that we do have very specific, if we remove the adjacent normal tissue for now, we're able to see very specific expression of these cytotoxic markers or these sort of effector cues within the cytotoxic T cell group. And again, this is something that we usually saw from one data set, but we're able to really compare this across 7 studies, for example, in this case. When we look at other markers here, what we're able to see is that there are very specific to like memory T cell resident markers. So we can see that unlike the cytotoxic population, these specific markers are more specific to these resilient memory T cell groups. And we actually are able to see other specific like FOXP3 expression within the sort of regulatory memory T cell sort of cell states as well. So these are things that we're able to really look at the expression within the lesional disease sampling site. We can add then another layer. Let's say we want to look at if there's any differences between lesional and adjacent normal tissue. We can apply that search, and that will automatically add that secondary filter. So we can see that currently from the studies, we only have 2 different sampling sites within the cytotoxic T cell group. And we do see some level of difference. So we do see that there are some markers like the NKG7, which is really an effector Q marker. And we really see these differences between adjacent and disease tissue. We can also see some other level of expression as well within the cytotoxic T cell that is not really seen in other populations. Okay, so that is something that you can start really understanding when you're looking at gene expression. Other questions that might come to mind is really, can I actually look at what specific studies are showing this? Is it something that only one data set has been able to find, or can I actually look at this sort of population at a study context? So within the expression by or these different settings, you're able to actually adjust not only to view the analysis, but you can look at this at a study context and really how these different genes are expressed or are shown across these different data sets. So this will show you if this is particularly only coming from one study, or can we really look at this enrichment profiling across these multiple studies as well? And what is the sort of percentage coverage? So how many cells are actually expressing this particular gene target within my specific cell population or sort of sampling site of interest. So this is really how you can get this sort of larger scale study scale understanding. You can also look at sample variability across multiple studies as well. within the public database. And especially when looking at these different cell types within these different lesion sites, you want to be able to see if this is consistent across multiple samples or really only coming from very specific samples that you might want to actually look even further into that data set and really understand what is the clinical context or what are the parameters that are used from those data sets specifically. So this is really how you're able to get this sort of from one study to this sort of multiple context and large scale analysis viewpoint. Okay, so if I go back to the analysis, another very important thing is how we're actually visualizing the data. So currently we're looking at a dot plot, which will give us sort of an initial context into the coverage or the level or average expression intensity score. But we are able to switch into the coverage heat map, which will give us more information on the actual percentage of those cells that are expressing within a particular cell type group or within that lesional site. So can we really profile these different sort of target markers? So that's another thing you can adjust as well as you're going through the platform. Now, one important question here, now that we've really answered, can we look at the expression and can we see local expression within the disease tissue that we don't see in sort of the adjacent normal tissue or specific differences between cytotoxic T cell expression as opposed to tissue resident memory markers? So from here, what we really want to try to also understand is, can we identify other genes that are co-regulated with our genes of interest within very specific disease context, but also sampling site. So looking at sort of the lesion site within that disease model and within that cell type group. So that's something that, again, we can go through our system. Let's say we've identified that there's a very interesting target that we see a very clear expression within the disease tissue and particularly within the cytotoxic T cell population that we don't see in the sort of tissue resident sort of cells. We can click on that specific gene within that label. So I'll remove the adjacent normal tissue because I'm really interested in the disease tissue context. And that will sort of update the plot. So what I want to see, is there any other markers that are co-regulated with this NKG7, particularly within our cytotoxic T cell population? So when we click on the gene, you'll see that it automatically sort of runs a Pearson correlation score where it's particularly looking at our cytotoxic T cell population within atopic dermatitis and particularly only our lesion sites. So this is where we can get a clear understanding as to what other co-markers are co-regulated within this context, and can we get more sort of information on how these sort of genes are sort of interacting, or what other sort of understanding can we gather from sort of cytotoxicity within the lesion site in dermatitis patients. So these are sort of information that you can gather. When you click on sort of the gene itself, you can run it across all the groups, or as I mentioned right now, you can run it within a very specific group. So in this case, we have our co-regulated markers for the cytotoxic population, but let's say we wanted to look at, in this case, we wanted to look at very specific co-regulated markers within memory regulatory T cells in the lesion site. We can then click on that specific target and that will rerun that sort of Pearson correlation score as well. So these are things that you can run in different contexts to really get this understanding on how these genes co-regulate and how that affects sort of inflammation or response to different treatments. So that's sort of the initial step. Other things that are quite important as well is really defining what are your signature markers within a very specific cell type population. So in this case, if we want to look at the specific signature markers for cytotoxic T cells across these data sets, and really looking at genes within the disease tissue, we can click on our cell type of interest that we want to look at even further, and that will run sort of a marker gene signature analysis. So you'll be able to define the top 100 sort of marker genes that are specific, in this case, to atopic dermatitis within the skin tissue structure, and also looking at different populations as well. So what are these core markers, and can I add these to the heat map to really look at these different sort of expression scores across these populations? Okay, so these are very features that are really important. This will allow you to also compare different groups. So if we want to run differential gene expression, you can use the specific analysis from here and go into this other sort of feature to run this direct comparison. And this is where you can add and use these different groups to really compare what are the genes that are differentially expressed across these data sets within the system. So not only a one study view, but doing DG analysis within multiple data sets. So this is something that you can add, for example, the anatomical structure. So I can add skin of body. So it particularly adds that across the data sets. And I want to add that across both groups. Okay, now here we can select our specific population. In this case, what I want to look at is the comparison within our tissue resilient memory T cell group within the disease tissue. So we can add that as a cell type category. And we can add our condition as well so that we make sure that the system will specifically look at atopic dermatitis. And we can do that across the other group as well. So now that we have our condition, we can add our cell type of interest, so our CD4 positive T cell. Okay, and our other group will be our cytotoxic T cell group. So we want to be able to compare these different cells and also within these different lesion sites as well. Okay, so now that we have here the sort of comparison, we can run the analysis, and that's when the system will be able to calculate the differentially expressed genes within the specific context or criteria. And this is again across 11 data sets, so we do have a larger number of cells than you would usually have in one study where you're looking at very specific cell populations as well. Okay, so that's sort of the initial analysis that you can perform on the Talk2Data system. There are other features like integrating these different studies and really looking at an integrated UMAP of the database. And that's something that you can also perform if you're planning to sort of integrate multiple studies within the database. Now, the next step, which was our other question, is really how can we take this even further where we're using sort of our AI Sonya agentic model to really answer other questions that are sort of related to looking at sort of lesion site differences, but also can we define and query a specific sort of biological question using natural language processing. So within AI Sonya, something that we need to understand is that there's different levels of sort of AI modeling. Normally what we want to really sort of gather or we want to sort of look for is models that really have this sort of agentic AI component. And this is really important, especially for drug discovery or looking at treatment response, because really speed only matters if these results are traceable and repeatable across the database. And there needs to be very specific ways where the agentic model communicates with the database, and it has different prompts, different types of sort of levels where it's trying to query that response, and it has different ways where these different agents are communicating these different prompts within the system. So this is really what we're trying to really try to focus on within our database, is really pulling in this agentic AI model and how it actually communicates directly with our public database across different types of data, not only single cell, but also spatial transcriptomics data. And really our goal is to get to the sort of different levels, not only sort of the core reasoning, where you're asking it how these different genes are expressed, but really going even further into these sort of multi-agent systems to really make this reproducible and validated across studies. So something that we'll see here in the Talk2Data agentic model is that we have different models for very specific reasons, whether that's for training it on biomedical research or customizing the AI model with in-house data, or really how can we integrate it with other systems or other sort of open AI targets or other sort of clinical trial sort of repositories as well. So that's something that we'll try to sort of show. So now that we are in sort of our system, we can go to the AI Sonya, and this is where we can ask very specific questions to the database. So some examples here are sort of more upper level, so they're not focusing only on atopic dermatitis, but particularly looking at inflammation and lesional sites and comparing that across different inhibitors within the public database. So when you're using the sort of search studies, it connects directly to the public database and it's able to really define what public data sets would be useful for this type of question. Do we have this in the database, or is it a study that would be useful for us to curate in the database? So that's one level of analysis you can perform in AI Sonya. The other levels is really where we're trying to actually answer very specific questions in the database. And in this case, let's say we were answering, let's say given that we are looking at CD4 memory resident T cells and cytotoxic T cells and atopic dermatitis lesions, can we confirm the expression of these specific genes and whether they're higher in memory resilient populations or cytotoxic populations? So that is something that you can ask the agentic model directly within the system. It'll try to process that request and run a sort of differential gene expression analysis directly from the system. So sort of how that actually connects, and you'll be able to see that there are different tools and parameters that the system um, allows to sort of have as an input when you're asking this sort of natural language processing query. And there's different ways where the system actually executes that. So there's multiple steps in the process where you have this AI agent communicating within the system. The actual tool executes an API query to the system to be able to look at the differential gene expression analysis. And then it actually summarizes that result for you and really what context or what output can we understand from this. So we can see here that there's different expression patterns within these genes, depending on the type of cell population and also the lesion sites within atopic dermatitis. So these are sort of questions that you can answer as well. If we're looking at how the system actually communicates with other systems or other sort of clinical trial databases, there are examples where you can ask the system, like, can we look at specific markers within open targets? And are there any within dermatitis or skin specific sort of conditions? So that's something that you can also ask within the system, where it will be able to run like a correlation Pearson score within these different markers, and it'll connect to the open targets database as well. So you'll be able to look at these different skin diseases. Are there any sort of targets, and can these relate to our condition of interest or have some description that we can use for further analysis as well? So there are very different questions that we can really interact, and these different cues and how it actually reasons that within the system and executes that through a specific API query. So this is sort of the other step I wanted to highlight as well. Now, if we go a step further and we identify that there's one particular study that has treatment data that has different lesion site sort of parameters, and we can look at the comparison across different cell type populations, we can then go into particular data sets in BBrowserX Pro to really look at this further. And particularly this study that came up within the initial filtering, we can actually see that this study has multiple treatment groups. It has different sampling sites and different sort of cell type populations that we want to look at further. And one of the questions that we really are trying to understand is, can we look at the ligand receptor pairs that are driving these cells to communicate and to cause inflammation or cause these differences in treatment response? So that's sort of a step further from the initial questions that we had from Talk2Data. So if we want to really look at the sort of ligand receptor shifts and how that relates to our populations of interest, so in this case, how these cytotoxic T cells might communicate with tissue resilient memory T cells, we can go into the database, so our specific studies tab, we can select that data set of interest that we want to look at further, and we can explore that study in BBrowserX Pro. So when we explore it, that study will open up in our other platform where we can perform very specific types of advanced analytical analysis, and we can actually look at very specific populations of interest. So in this case, our main question is, can we identify these ligand receptor pairs that are driving our specific cells to communicate? And is this different across different conditions or different treatments? So when we run the cell-to-cell communication, you can sort of concentrate on very specific populations. In this case, I'm including these different memory sort of resident T cell groups, regulatory T cells, and also our cytotoxic T cell populations. I want to look at the differences in different conditions, so I can input that as a metadata reference, and then I can run it. So when we look at cell-to-cell communication, this is where we'll have these direct comparisons between atopic dermatitis, specific lesion sites, and also the healthy adjacent tissue coming from healthy controls. So we can see that there's very specific differences where our cytotoxic and tissue resilient cells are actually communicating within the atopic dermatitis. And this is something that we don't see in the healthy control tissue. So if we want to look at these specific cells and how they communicate in this case, we can click on this specific sort of receiver and sender pair, and we can look at the specific ligand binding receptors that are specifically driving this communication within atopic dermatitis. When we want to select a specific interaction, we can select these specific to our disease of interest, and we can apply that interaction search. So that will allow us to really look at those ligand means and receptors that are really driving these cells to communicate. In this case, are cells of interest. And we can look at this across these different ligand receptor pairs as well. So this is really how you can get a more sort of further understanding. And we can see here that in this case, atopic dermatitis lesions really have very specific amplification on how these T cells communicate. We do see that there's sort of higher tissue resilient sort of memory T cells that are sort of receiving that information, and then they're communicating with the cytotoxic populations as well. And this is really sort of leading to these really strong T to T cell signaling responses within atopic dermatitis. Okay, we can also see selective checkpoint inhibitory access, particularly when we're looking at CD161, positive memory, sort of tissue resilient memory T cells, and also this effect that is not only global to T cells as well. So really, this is sort of other analysis that we can gather from BBrowserX Pro when we're looking at one individual public data set. Okay, so now that we finalized this, I just wanted to summarize some of the results that we were able to look at. So within the Talk2Data database, we were able to show how we can really perform different types of filtering criteria when it comes to the tissue structure, the type of disease model, and also different lesion or adjacent sampling sites, how we can query very specific markers and define these populations within sort of local expression. We also perform differential gene expression analysis across the entire data sets within the system based on our criteria, and we defined very specific markers that were either cytotoxic, enriched, or that were enriched within the sort of tissue resident T cell groups. The cell-to-cell communication analysis showed really strong signaling across different T subsets, and particularly ligand binding receptor pairs like CLE, C2D, KLRB1, which were very specific interactions leading to this sort of inflamed skin region. So these are really inputs that are really important when you're trying to really understand what is happening within this sort of atopic dermatitis lesion sites or the microenvironment within the skin? And can we target these and look at these different sort of cell type populations that drive either inflamed tissue or that respond in many different ways within local expression? Okay, so now that we're done, we can gather some feedback from the users that join the call. So there will be either a poll or questions that we can gather if these sessions are really useful, if these are features that you would normally use within your own analysis, or is there any other sort of specific features that you think would be very important to integrate within the Talk2Data system that you didn't really see today? So please, if you have some time, can you fill out the sort of poll, and that will give us more understanding into your own workflow and how can we incorporate that in the database. Okay. So we'll give a few minutes for that to be completed. While that's completed, I'll just check if there's any questions within our chat that might be really important to answer within the users who joined today. Okay, so I don't see any particularly open questions. If you do have a question, feel free to add it in the Q&A chat, and we're happy to sort of clarify that as well within the system. Okay. So I think this is sort of the session for today. So we've reached sort of the end. I hope this was useful for you to get an understanding how you can utilize Talk2Data in this different sort of context, looking at sort of skin lesions. But again, this can be applied to many different disease contexts. So if you are interested in really understanding how this might work for your specific use case, you can always reach out to our team and we can do a tailored sort of training session with you where we go through these questions and how the system would be able to answer these. So thank you everyone who joined today, and we look forward to seeing you in the next session as well. Thank you so much.